Hi everyone, welcome to Lecture 10C, the third lecture for week 10 of Applied Immunology and the last lecture for this entire course. Today we will build off of what we learned about tumor cells in Lecture 10B by summarizing a powerful newer class of anti-cancer drugs called immunotherapies. We will start off by talking about the traditional types of cancer treatments and how immunotherapy can offer some improvements where some of these fall short, and then summarize the mechanism of action of several different categories of cancer immunotherapies. We will then end by discussing some of the drawbacks of immunotherapy treatments and quickly go over some future directions for this field that will be crucial for optimizing the efficacy of these drugs. Please note that the discussion prompt for week 10 relates to summarizing a given cancer immunotherapy, so if you plan on responding to the final discussion board this week, then this should be a particularly helpful lecture. I think it's helpful to start off our discussion of cancer immunotherapy by considering where it fits in with more conventional forms of cancer treatment. Clinicians have historically referred to these as the three pillars of cancer therapy. These include surgery, or the physical removal of a tumor mass, application of radiation, which can damage DNA in rapidly dividing tumor cells, which forces them to undergo apoptosis, and lastly, chemotherapy, which is the administration of toxic drugs that will kill off rapidly dividing cells, such as tumor cells. However, there are drawbacks to using each of these types of treatments. Surgery can remove the majority of a tumor mass, but will almost always leave behind some portion of cancer cells that can regrow a new tumor later on. Radiation and chemotherapy kill off tumor cells, but they also have major off-target effects with killing other rapidly dividing cells in the body, and these include bone marrow stem cells, as well as epithelial cells at mucosal surfaces. I hope that most of you haven't had someone close to you have to go through chemo and radiation, but if you do, then you probably understand how awful some of these side effects can be, since you're essentially poisoning someone's body in an attempt to kill off all of their tumor cells. Aside from the off-target effects of chemo and radiation, another issue with these treatments is the fact that they apply what we can view as a pretty static form of therapy against what is an evolving set of cancer cells. From our last lecture, we discussed the concept of immunoediting and how this selects for tumor cell clones that can escape immune detection. And we can think of the exact same process working in response to these three pillars of cancer treatment. Application of radiation or chemotherapy will oftentimes select for tumor cell clones that are resistant to that particular therapy. So if you keep giving a patient the same treatment over and over again, you're likely to end up with a resistant tumor in the end. The main advantage of immunotherapy compared to some of these other forms of cancer treatments is that the immune system is relatively adaptable, which is really important when you're trying to overcome a disease as nasty as cancer, where transformed cells have been selected for all sorts of different strategies to promote their own survival. This adaptability is due in part to the fact that many different immune cells can be used to target and help kill a transformed cell. And importantly, within a pool of cells like T cells, there are many different clones with differing specificities. Say that you have a cancer cell clone X, which can be killed by an X-specific T cell. This essentially exerts a selection pressure on the tumor for clones or variants that are no longer detected by X-specific T cells. So you get variants like cancer cell clone Y that have evaded that first mechanism of tumor control. However, because adaptive immunity encodes many different specificities, there's likely a Y-specific T cell that can now eliminate that escape clone. And this is the entire premise of the tumor-immune interactions that occur during the equilibrium phase of tumor growth. We know that the endogenous immune response cannot accomplish this with 100% efficiency. Otherwise, nobody would develop cancer, and we know that this obviously is not the case. The strategy of cancer immunotherapies is therefore aimed at enhancing the ability of tumor-specific immune cells to engage these responses against what we can view as a moving target. And this is in order to overcome mechanisms of tumor immune suppression that we learned about in Lecture 10B. Importantly, immunotherapies must accomplish this in a way where all tumor cells are successfully eliminated before the tumor has a chance to become fully resistant to all immune mechanisms of elimination. So we can think of these types of drugs as working by boosting the endogenous responses involved in cancer immunosurveillance, so that they're more effective in eliminating tumors before immune editing selects for an escape variant. There are many different types of cancer immunotherapies that try to accomplish this, and each of these functions by targeting different aspects of an immune response. Many of these strategies try to address the immunosuppressive tumor microenvironment that we talked about in the last lecture. Modulation of the tumor microenvironment may involve introducing more pro-inflammatory signals or depleting immunosuppressive cell subsets. In addition to addressing the tumor microenvironment, other immunotherapies are designed with the goal of reinvigorating tumor antigen recognition, which will stimulate adaptive immunity. We can subdivide immunotherapies based on whether they accomplish these goals by targeting either innate or adaptive immune responses. Let's start with immunotherapies that primarily target the innate immune system. These include the administration of inflammatory cytokines that can counteract the immunosuppressive tumor microenvironment, including things like type 1 interferons or IL-12. This favorable manipulation of the tumor microenvironment is also addressed through administration of inflammatory ligands that can stimulate pattern recognition receptors like toll-like receptors or CGAS sting sensing, and these function by promoting inflammatory activation of innate immune cells, which then in turn induces things like cytokine and chemokine production along with antigen presentation. Lastly, several immunotherapies target macrophages because tumor macrophages are usually quite tolerizing or tolerogenic, and they're associated with adverse cancer prognoses. Monoclonal antibodies can be used to block immunosuppressive signaling in macrophages through something called the TAM receptors, or promote tumor phagocytosis by blocking this other molecule called CD47. Monoclonal antibodies can also be used to deplete macrophages through the mechanisms that we discussed in week 6, although most of these don't have great specificity for tumor macrophages themselves, and so they can still affect other macrophage populations throughout the body. For the sake of time, we won't discuss much more of these innate immunotherapies, though you have the option to do some reading on these for the discussion prompt this week if you're interested in learning more. Immunotherapies with some of the best clinical responses that we've seen typically involve the manipulation of adaptive immunity, particularly by augmenting CD8 T cell function, since this cell type is so critical for anti-tumor responses. We will talk about some of these in more detail for the remainder of the lecture, but let's go over a quick summary first. First off, hematopoietic stem cell transplants from allogeneic donors are often used to treat certain types of leukemia patients that are non-responsive to traditional radiation and chemotherapy. Leukemias are blood cancers that can involve things like B-cell or myeloid cell malignancies. So irradiating and killing off the defective bone marrow to replace it with transplanted stem cells is a good way of treating disease, particularly if you get a beneficial graft versus leukemia effect that we talked about back in week 9. This is kind of a different example since it doesn't reactivate the immune system per se, but I still wanted to include it here since it's used to treat this subset of cancers or leukemias. Immunotherapies that truly function by reinstating responses to tumor antigens include cancer vaccines, which deliver defined tumor antigens in a way that stimulates tumor-specific T cell responses. Monoclonal antibodies are another type of drug that can be used to either directly kill tumor cells, deplete immunosuppressive cell subsets, such as Tregs, or modulate T cell activation status through things called checkpoint blockade and co-stimulation. Lastly, immunotherapy has yielded a new class of cell-based products that are made using T cell engineering. These T cells are isolated from a cancer patient, manipulated or engineered ex vivo to express any of a number of beneficial receptors or signaling molecules, and then reinfused back into the patient through a process called adoptive transfer. Let's learn a little bit more about some of these immunotherapies that target tumor-specific adaptive responses, starting off with cancer vaccines. These vaccines induce tumor-specific T cell responses in patients that already have cancer. They essentially operate with the same goal as vaccines designed to prevent infectious disease, so they introduce a specific antigen in a non-dangerous way that can boost antigen-specific adaptive immunity. But in this context of cancer vaccines, obviously the antigen is one that's expressed by tumor cells rather than a pathogen. Because cytotoxic T cells are so important for defense against tumors, we primarily evaluate the efficacy of cancer vaccines by their ability to stimulate antigen-specific CD8 responses. There are a few different ways to make cancer vaccines, but the one that we will focus on today is something called a dendritic cell-based vaccine, which means that you engineer dendritic cells to present a specific tumor antigen on MHC to stimulate T cells. The best example of an effective cancer vaccine is something called Provenge, which can be used to treat metastatic prostate cancer. Making this type of dendritic cell-based vaccine is a bit more complicated than some of the vaccine formulations that we learned about back in week 8. For dendritic cell vaccines, you start off by isolating peripheral monocytes from the blood of the prostate cancer patient. These are then cultured ex vivo with a fusion protein that contains both a prostate cancer antigen called prostatic acid phosphatase, or PAP, and this is coupled to GM-CSF, which is a growth factor that induces dendritic cell differentiation from monocytes in blood. This fusion protein is processed by the monocyte so that GM-CSF triggers dendritic cell differentiation. And since the cells are loaded with PAP antigen, they process this antigen and present it on MHC. The resulting tumor antigen-loaded DCs are then reinfused back into the same patient. This is something called an autologous cell transfer, because a patient's own cells have been isolated, modified, and then reinfused back into the same patient. Autologous cell transfers have the benefit of not causing any transplantation rejection issues related to mismatch in histocompatibility antigens. And we will talk about these again in a little bit when we cover T cell engineering. For this case of Provenge, once the PAP-loaded autologous DCs are reinfused, they can then prime PAP-specific T cell responses that lead to CD8-mediated prostate cancer killing. An obvious drawback to cancer vaccines is that they work best for tumors with strictly defined tumor antigens that are conserved amongst all tumor cells. This is actually the case with PAP expression in prostate cancer, but many spontaneous cancers have more heterogeneity in terms of their antigen expression profiles. Another type of immunotherapy based on defined tumor antigens are monoclonal antibodies that recognize a given tumor antigen. These types of antibodies can be used to kill tumor cells in a number of ways. So first, a tumor-specific antibody by itself can bind to a tumor antigen. This tags the tumor cell for killing through mechanisms like NK cell-mediated antibody-dependent cytotoxicity, which we've learned about previously. Examples of immunotherapies that work this way include Herceptin, which recognizes HER2, which is an antigen that's overexpressed in some breast and ovarian cancers. There's also an antibody called rituximab, and this is an anti-CD20 monoclonal that has worked well in the treatment of non-Hodgkin's B-cell lymphomas. Antibodies can also be conjugated to various types of cytotoxic payloads, in which case they function by targeting the delivery of cytotoxic molecules to antigen-expressing tumors. This is usually done using a single FAB fragment that contains the antigen-specific variable region of an antibody, which is conjugated to a cytotoxic molecule. The advantage of using these FAB fragments instead of whole antibodies is since the fragments are smaller, they are better at penetrating into a tumor mass and then being taken up by tumor cells. The cytotoxic molecules linked to an antibody fragment in these situations include bacterial toxins, chemotherapy agents, or radioisotopes, all of which function by killing the tumor cell that's bound to the FAB fragment. The middle and right panels of this figure show a few different examples of antibody-drug conjugates. Like with cancer vaccines, the efficacy of these tumor-specific antibodies relies on the identification of a tumor antigen. So unfortunately, these forms of cancer immunotherapies cannot be used for cancers that are poorly immunogenic or those that express unknown tumor antigens. However, monoclonal antibodies can also be used as immunotherapies for tumors with poorly defined antigens. In this case, rather than targeting tumor cells directly for killing, this class of monoclonal antibodies functions by enhancing the efficacy of pre-existing T cell responses that have been mounted against tumors. To understand how these work, we need to look at some of the different co-stimulatory signals and receptors that can influence T cell activation. We learned about these in our week 4 and week 8 lectures on T cell activation and the resolution of T cell responses. but let's do a quick refresher on some of the relevant signaling molecules here. Upon TCR recognition of peptide MHC, we know that this relays an activating signal 1 back to the T cell. However, the T cell cannot become fully activated unless it also receives a co-stimulatory signal through a receptor like CD28, which recognizes CD80 or CD86 expressed by the APC. This is considered signal 2 of T cell activation, and we're used to thinking about it being produced by APCs that are presenting peptide to initiate effector T cell activation. We haven't covered some of these molecules yet in this course, but there are actually several other stimulatory receptors besides CD28 that can help activate T cells. These include CD137 and OX40, which can bind to their corresponding ligands that are expressed by APC. Back in week 8 of this course, we learned that there are also a number of receptors expressed by T cells that can relay an inactivating form of signal 2. These include surface markers like PD1, TIM3, and CTLA4 at the top of this diagram, all of which can bind to ligands expressed by APCs or even tumor cells themselves. Activation of these inhibitory receptors induces a non-functional state in effector T cells. Note that CTLA4 also binds to CD80 or CD86, so it basically competes with an activating CD28 for ligand binding. These receptors are sometimes referred to as immune checkpoints, since they serve as a check or regulation on T cell activation. Monoclonal antibodies that bind to these two different types of co-stimulatory receptors can be used to essentially reinvigorate T cells that have been tolerized against a tumor. The first strategy of doing this is by using monoclonal antibodies that inactivate inhibitory receptors, which is the case with antibodies against PD1 or CTLA4. These antibodies have been quite successful when used in the clinic against certain types of tumors because they essentially de-repress T cell inhibition. You may have also heard of these antibodies referred to before as immune checkpoint blockade. Monoclonal antibodies can also do the converse of this, where they function by binding to stimulatory receptors in a way that activates them. This is the case with monoclonals against CD137 or OX40, which work by overriding any inhibitory signal 2 relayed to the T cell by either tumor cells or tolerogenic APCs that have been induced by immunosuppressive tumors. These types of monoclonal antibodies are advantageous in that they essentially restore the functionality of pre-existing T cell responses against tumors, so they do not require identification of tumor antigens. The last type of immunotherapy we will talk about today are some really powerful T cell products that have been engineered to optimize their ability to identify and kill tumor cells. This is often referred to as adoptive T cell therapy and consists of a similar process to the Provenge dendritic cell vaccine that we talked about a few slides ago. Here, you instead isolate circulating T cells from a cancer patient and then manipulate them in vitro, or you engineer them to either express or knock out any of a number of possible targets. Once these cells are successfully modified, they are expanded to high numbers using in vitro cell culture and then reinfused back into the patient. Again, because these cells originated in the same patient that they eventually go back into, they are referred to as autologous engineered T cells. The goal of this therapy is to introduce modifications using T cell engineering, which make the T cells better at their effector functions. And this would then allow them to successfully eliminate all cancer cells in the patient to try and cure them of disease. Considering how many ways tumors can evade immune responses, you can use T cell engineering to generate T cells that would override pretty much any one of these mechanisms of immune avoidance. This includes overexpression of pro-survival or inflammatory cytokines to overcome the immunosuppressive tumor microenvironment. You can also express chemokine receptors that would promote T cell migration into a tumor, or you can knock out regulatory genes that dampen T cell activation. Lastly, you can modify patient T cells to express synthetic versions of antigen receptors that are better suited for T cells to recognize and kill tumor targets. And this is a very important component or subtype of T cell engineering. One example of these modified antigen receptors is something called a chimeric antigen receptor, or CAR. CAR engineering has yielded many different types of these receptors, But this example looks at a CAR with specificity against a CD19 antigen, which is expressed by B cells. These receptors usually contain an extracellular domain that's shown here in red, which is essentially the region of an immunoglobulin molecule that recognizes CD19 that's expressed on the surface of a cancer cell. Now, these are called chimeric antigen receptors because this extracellular antigen recognition domain is then fused to intracellular domains that resemble the signaling machinery of the T cell receptor. This usually includes a CD3 zeta chain and a co-stimulatory domain called 41BB, and each of these contain ITAMs that are needed to relay T cell signal transduction. The advantage of these chimeric receptors is that engineered T cells expressing the CAR can become activated in response to antigen recognition alone. And importantly, since the antigen recognition domain of these receptors functions more like the variable region of an immunoglobulin molecule, the T cell no longer has to recognize antigen that is presented on an MHC. So the use of these chimeric receptors removes a lot of the restrictions on T cell activation. This is a similar schematic to the one shown on the last slide that outlines the process of autologous T cell engineering to express this given CD19 CAR in T cells. In this example, these T cells would be taken from a cancer patient that suffers from some form of B cell malignancy, and B cell leukemias and lymphomas respond well to CAR T cell engineering, as patients who receive CD19 CAR T cells exhibit pretty high rates of complete remission from their cancer. This is impressive, especially when considering that most of these patients that receive CD19 CAR T cell therapy have already developed leukemias that are resistant to conventional therapies like chemo and radiation. Some limitations do exist to CAR T cell therapy, and these include the targeting of other healthy cells that share antigen expression with the tumor cells. In this example of using a CD19 CAR T cell, healthy B cells in the leukemia patients are also killed off by this therapy. Another issue with CAR T cells is that at least some subset of patients exhibit antigen escape. So in this example, this would involve some leukemia cells that internalize or lose expression of CD19. So these are selected for outgrowth following immunotherapy. Despite these issues, the field of CAR engineering opens up a lot of exciting possibilities for optimizing T cells used to fight tumors. Despite the advantages of immunotherapy, there are some dangerous off-target effects that are observed in a subset of patients. One of these is uncontrolled systemic inflammation following administration of CAR T cells to treat things like B-cell leukemia. And these effects are thought to actually result as a product of how well this therapy works, since cancerous B cells throughout the body are suddenly being attacked all at once following infusion of CAR T cells. These inflammatory pathologies look a bit like sepsis and are sometimes referred to as cytokine release syndrome or tumor lysis syndrome, since they are often characterized by high levels of circulating inflammatory cytokines. You can see from this graph on the left that increased serum levels of IL-6 and interferon gamma in these patients correlates with higher grades of more severe clinical symptoms. IL-6 levels are also correlated with severe neurological toxicities, which are shown by this figure on the right. These are extremely dangerous side effects that can kill patients, so it's crucial to monitor people that receive CAR T cell therapy so that they can receive interventions if necessary. Another major effect of immunotherapies is that some of these can break peripheral tolerance and lead to forms of autoimmunity. This has been observed with immune checkpoint blockades such as anti-CTLA-4 or anti-PD-1. These scans are from a patient with stage 4 metastatic melanoma who had several lung metastases, as shown by the black arrows in the left figure, and these were eliminated following co-administration of radiation along with anti-CTLA-4. However, this patient developed severe colitis soon after treatment, and examination of a colon biopsy revealed that they had massive T-cell infiltration into their gastrointestinal tract, and this is shown by the brown staining for the T-cell marker CD3 on the right image. Other immunopathologies that are developed following immune checkpoint blockade include severe dermatitis, liver damage, and even type 1 diabetes. And this really highlights the importance of T-cell checkpoints in continually maintaining peripheral tolerance to self-antigen. With autoimmune pathologies in particular, it's important to keep in mind that many cancer patients who get put on immunotherapy regimens may have tumors that have become non-responsive to all other forms of conventional cancer therapy. In this situation, even if autoimmunity develops, immunotherapy may represent someone's last available treatment option before they have to go into palliative hospice care. If autoimmune symptoms are things that could be managed by medication, then most people would consider these risks to be worth it for another chance at survival. With that being said, there's clearly lots of work to be done in order to optimize treatments in a way that decreases the risk of these rather severe side effects. Aside from minimizing off-target toxicities, there are a few other next steps for the development of cancer immunotherapies. The T-cell engineering strategies that we went over today all involved cases of autologous cell transfer, where cancer patients' own T-cells are removed, modified, and then reinfused. This entire protocol is highly personalized, which means that it's expensive, time-consuming, and only available to people that have access to advanced medical care. Next-generation engineered T-cells will include ones that are modified from a single donor, which could then be administered to many different allogeneic recipients. These are referred to as off-the-shelf CAR T-cells or universal T-cells, which would be mass-produced and therefore a lot cheaper and more accessible compared to autologous T-cell engineering. Another frontier for tumor immunotherapy is to get these drugs to work well in solid tumors. Things like CAR T-cells can work amazingly well for leukemias where the tumor cells are circulating freely through the body, so it's easier for a T-cell to access an individual tumor cell. However, a lot of immunotherapies don't work as well in solid tumors, since these can contain a whole other set of harsh immunosuppressive mechanisms that we talked about in the last lecture. Testing new strategies for boosting the efficacy of immunotherapy in solid tumors is an important area of ongoing research. Other future directions include determining how to screen patients that are likely to respond to cancer immunotherapy. This is really important in terms of not putting cancer patients through treatment regimens that are unlikely to work for their type of cancer. The amount of novel tumor neoantigens expressed by a tumor, which is also called the tumor mutational burden, appears to be an important correlate of favorable responses to immunotherapy, but more research needs to be done in this area as well. Lastly, tumor immunotherapies tend to be extremely expensive, which again brings up an issue of treatment accessibility to all patients. Cancer treatments can be very expensive just to begin with, but high doses of monoclonal antibodies and personalized engineered T-cells can really drive that cost through the roof. So even if a patient survives cancer, they may then be saddled with serious medical debt. This is a big problem with respect to equity in healthcare, so I think it's an important thing to note, especially for those of you that are interested in careers in clinical medicine. There's been a lot of hype over immunotherapy in recent years, given how well things like CAR T-cells and immune checkpoint blockade have worked in certain types of cancers. However, I think that the most realistic way to view immunotherapy is that it represents a fourth pillar of cancer treatments, that rather than serving as a magic bullet that completely replaces more conventional treatments, can be added to the repertoire of available tools that can be used to treat cancer patients. It's likely that long-term cancer remission in many patients would result from immunotherapy used in some combination with other cancer treatments. Cancer is a difficult target to eliminate, but the rational selection of therapy combinations that include immunotherapy will hopefully lead to some improved survival outcomes for patients that suffer from this really terrible type of disease. I want to end today by trying to connect what we've learned today about cancer immunotherapy with some of the other pathologies that we've covered in the second half of this course. I think that this is most easily done by considering this diagram we've visited a few times in recent weeks, which represents the balance that must be struck between inflammatory immune responses and tolerogenic immune responses. In the context of tumor immunotherapy, we're really shooting for this sweet spot in the middle, represented by the black arrow, where there's enough inflammation and immune activation to fully eliminate a tumor, but not too much inflammatory immune activation that it could lead to cytokine release syndrome or autoimmune pathologies. The balance between protective inflammation versus immunological tolerance is something that has come up in pretty much every disease context that we've covered in this class. From our first lecture earlier this week, we learned that at mucosal surfaces, the immune system must maintain this balance by being able to respond to invasive pathogens while preventing inflammatory responses mounted against harmless commensal microbes or food antigens. Allergic reactions, or hypersensitivities, occur when protective responses that are usually directed towards harmful pathogens are instead activated in response to harmless environmental antigens called allergens, and this leads to inflammatory pathologies. Autoimmune diseases occur when tolerance to self-antigen is broken, meaning that the inflammatory responses used against infectious microbes are mistakenly directed towards self-tissues. Any therapies used to treat autoimmunity must try to restore this balance between inflammation and tolerance in order to suppress pathological immune activation, again, while still maintaining the ability to fend off infection. Lastly, recipients of allogeneic organ transplants must also receive treatments that suppress immune responses enough to prevent allograft rejection while still maintaining the ability to eliminate pathogens. Across each of these disease settings, therapeutic attempts to restore this immunological balance can be extremely difficult, and it's likely that any patient will have some adverse side effects on one side of the scale or the other. We've covered a lot of information and immunology jargon throughout the 10 weeks of this course, so I wanted to make sure that we end this final lecture on a more conceptual note, where we focus on how the balance between inflammation and tolerance is a recurring theme in any disease that has an immunological component. Keeping this in mind is important in deciding how to treat these pathologies from a clinical standpoint, as well as in thinking about how these diseases and therapies might mechanistically function from a research standpoint. To summarize, today we've introduced a class of anti-cancer treatments called immunotherapies, which can work using several mechanisms to improve immune recognition and elimination of transformed cancer cells. Although these can be added for use in combination with existing pillars of tumor therapy, they still need to be optimized in order to minimize off-target side effects. We went over a quick summary of innate and adaptive cancer immunotherapies and spent a little extra time explaining how things like tumor vaccines, monoclonal antibodies, and CAR T cells function. Although these treatments show some really amazing tumor remission responses for patients with certain types of cancers, there's still a lot of room for improvement. Optimizing these drugs to have fewer off-target toxicities, to have better efficacy in solid tumors, and improving pipelines for production that address accessibility and health equity concerns are all important areas to improve for next-generation immunotherapies. Lastly, we ended today by trying to connect how the fine balancing act between protective inflammation and immunological tolerance applies to cancer immunotherapy, along with the other types of diseases that we've learned about in the second half of this course. This is the last lecture for week 10, so congratulations on officially finishing all of the lecture materials for Applied Immunology. Please check Canvas for accompanying reading assignments for this week, as well as your last extra credit quiz and your last opportunity to submit a graded discussion response, again, covering a cancer immunotherapy of your choice. Otherwise, the only assignment that you have left is the final exam for Applied Immunology, which will be due at the end of week 11, so please check Canvas for course announcements related to the final. Good luck with your remaining work for this course.